Dear friends, hello and welcome. Today I am going to deliver a lecture on extra chromosomal inheritance and chloroplast variegation in 4 o'clock plant and Chlamydomonas. The objectives of today's lecture are: number first, to give a general and thorough idea about extra chromosomal inheritance. Number second, to give a brief introduction about 4 o'clock plant and Chlamydomonas. Number third, to discuss chloroplast variegation in 4 o'clock plant. And finally, we shall be discussing chloroplast variegation in Chlamydomonas. Now, we will start with the extra chromosomal inheritance. The speculation that chromosomes might be the key to understanding heredity led several scientists to examine Mendel's publications and re-evaluate his model in terms of the behavior of chromosomes during mitosis and meiosis. In 1902, Theodore Boveri observed that proper embryonic development of sea urchins does not occur unless chromosomes are present. That same year, Walter Sutton observed the separation of chromosomes into daughter cells during meiosis. Together, these observations led to the development of the chromosomal theory of inheritance, which identified chromosomes as the genetic material responsible for Mendelian inheritance. Despite compelling correlations between the behavior of chromosomes during meiosis and Mendel's abstract laws, the chromosomal theory of inheritance was proposed long before there was any direct evidence that traits were carried on chromosomes. Critics pointed out that individuals had far more independently segregating traits than they had chromosomes. It was actually only after several years of carrying out crosses with the fruit fly Drosophila melanogaster that Thomas Hunt Morgan provided experimental evidence to support the chromosomal theory of inheritance. Extranuclear inheritance refers to inheritance patterns involving genetic materials outside the nucleus. The two most important examples are due to genetic material within the organelles, that is, mitochondria and chloroplasts. These organelles are found in the cytoplasm. Therefore, extranuclear inheritance is also termed as cytoplasmic inheritance. In other words, extranuclear inheritance occurs when phenotypes result from genetic influence other than the biparental transmission of genes located on chromosomes housed within the nucleus. Organelle heredity, an example of extranuclear inheritance, is due to the transmission of genetic information contained in mitochondria or chloroplasts, most often from only one parent. Extrachromosomal inheritance is the inheritance of traits through DNA that is not connected with the chromosomes but rather to DNA from organelles in the cell. The genes governing cytoplasmic inheritance are called plasma genes or cytoplasmic genes, cytogenes or extranuclear genes, while the sum total of extrachromosomal genetic material is known as plasmon. Simons in 1921 suggested that the term idiotype should be used to denote the sum total of nuclear genes, while the extrachromosomal genes should be called plasma type. Extranuclear genes display non-Mendelian inheritance, which has four characteristics. Number first, typical Mendelian ratios do not occur because meiosis-based segregation is not involved. Number second is the reciprocal crosses usually show uniparental inheritance. Number third, extranuclear genes cannot be mapped to chromosomes in the nucleus. And number fourth, if a nucleus with a different genotype is substituted, non-Mendelian inheritance is unaffected. Now, let us discuss the characteristics of cytoplasmic inheritance. Number first is the reciprocal differences. Reciprocal crosses show marked differences for the characters governed by plasma genes. In most cases, plasma genes from only one parent, generally the female parent, are transmitted, and this phenomenon is known as uniparental inheritance. Number 2 is the lack of segregation. In general, F2, F3 and the subsequent generations do not show segregation for a cytoplasmically inherited trait. This is because the F1 individuals generally receive plasma genes from one parent only. Number 3 is the irregular segregation in biparental inheritance. In some cases, plasma genes from both the parents are transmitted to the progeny. This is known as biparental inheritance. Number 4 is the somatic segregation. Plasma genes generally show somatic segregation during mitosis, a feature of rare occurrence in the case of nuclear genes. Number 5 is the association with organelle DNA. Several plasma genes have been shown to be associated with chloroplast DNA or mitochondrial DNA. Number 6 is the nuclear transplantation. If nuclear transplantation reveals a trait to be governed by the genotype of cytoplasm and not by that of nucleus, cytoplasmic inheritance of the trait is strongly indicated. In nuclear transplantation, nucleus of a cell is removed and placed by a nucleus of another genotype from a different cell. Generally, nuclei of somatic cells are transplanted into zygotes before the first meiotic division is initiated. Number 7 is the transfer of nuclear genome through Bragg crosses. The nucleus of a variety of species may be transferred into the cytoplasm of another species or variety through repeated back crossing with the former, which is used as a recurrent male parent. Lines produced in this way are known as alloplasmic lines since they have nuclei and cytoplasms from two different species. Number 8 is the mutagenesis. Some mutagens, for example, ethidium bromide, are highly specific mutagens for plasma genes, while nuclear genes are not affected by them. Induction of mutation by such agents in a gene indicates it to be a plasma gene. Number 9 is the lack of chromosomal location. In many organisms, extensive linkage maps of nuclear genes are available. If a gene is shown to be located in one of these linkage groups, it cannot be a plasma gene. Failure to demonstrate the location of a gene in one of the linkage groups of an organism is indicative of its cytoplasmic location, but this is highly tentative. Number 10 is the lack of association with a parasite, symbiont or virus. In many cases, a cytoplasmically inherited character is associated with a parasite, symbiont or virus present in the cytoplasm of the organism. Such cases cannot be regarded as cases of cytoplasmic inheritance. Only those cytoplasmically inherited characters which are not associated with parasites, symbionts or viruses can be regarded as governed by plasma genes. The known cases of true cytoplasmic inheritance are concerned with either chloroplast or mitochondrial traits and are usually associated with their DNA. Such cases are therefore often referred to as organellar inheritance, plastid inheritance and mitochondrial inheritance. Many geneticists have studied various cases of extranuclear inheritance in different eukaryotes. Number 1 is the maternal inheritance. In certain cases, it has been observed that certain characteristic phenotypic traits of F1, F2 or F3 progeny are not the expression of their own genes but rather those of their maternal parents. Such phenotypic expression of maternal genes may be short-lived or may persist throughout the lifespan of the individual. The substances which produce the maternal effects in the progeny are found to be transcriptional products of maternal genes which have been manufactured during oogenesis and which exist in the ooplasm of unfertilized eggs in the form of inactive protein coated and late translating mRNA molecules or inactivated rRNA and tRNA. Number 2 is the extranuclear inheritance by cellular organelles. Chloroplast and mitochondria are organelles that contain their own DNA and protein synthesizing apparatus. A widely held theory concerning their origin proposes that they were once infectious endosymbiotic prokaryotes that evolved such a dependence on the gene products of the host that they are no longer able to function autonomously. This theory has been supported by the fact that the genetic components of these organelles are often similar to those found in prokaryotes. Number third is the extranuclear inheritance by endosymbiotes. Certain intracellular parasites such as bacteria and virus particles maintain symbiotic relationship with host cells. They are self-reproducing and look like the cytoplasmic inclusions. Sometimes they exhibit an infection-like transmission with a hereditary continuity of their own. Generally, such symbiotes are coined by letters of the Greek alphabets, that is sigma, kappa or mu, etc. Number fourth is the uniparental inheritance in Chlamydomonas. Like fungi, algae rarely have different sexes, but they do have mating types. In many algal and fungal species, there are two mating types that are determined by alleles at one locus. A cross can occur only if the parents are of different mating types. The mating types are physically identical but physiologically different. Such species are called heterothalic. Now, let us try to have a look at 4 o'clock plant and Chlamydomonas. Mirabilis jalapa, that is the 4 o'clock flower, is the most commonly grown ornamental species of Mirabilis and is available in a range of colors. Mirabilis in Latin means wonderful and jalapa is a commonplace name in Central and North America. A curious aspect of this plant is that flowers with different colors can be found simultaneously on the same plant. Additionally, an individual flower can be splashed with different colors. Flower patterns are referred to as sectors, that is whole sections of flowers, or flakes, that is stripes of varying length, and spots. A single flower can be plain yellow, pink or white, or have a combination of sectors, flakes and spots. Furthermore, different combination of flowers and patterns can occur on different flowers of the same plant. Another interesting point is a color changing phenomenon. For example, in the yellow variety, as the plant matures, it can display flowers that gradually change to a dark pink color. Similarly, white flowers can change to light violet. Now, Chlamydomonas. Chlamydomonas is a genus of unicellular green algae that belongs to Chlorophyta. These algae are found all over the world in soil, fresh water, oceans and even in shallow or mountain tops. Algae in these genes have a cell wall, a chloroplast, an eye that perceives light and two anterior flagella with which they can swim using a breaststroke type motion. More than 500 different species of Chlamydomonas have been described, but most scientists work with only a few. The most widely used laboratory species is Chlamydomonas reinhardti. Cells of this species are haploid and can grow on a simple medium of inorganic salts using photosynthesis to provide energy. Now, let us discuss chloroplast variegation in 4 o'clock plant. One of the classical examples of extrachromosomal inheritance is the variegated leaves found in the 4 o'clock plant. There are 3 kinds of phenotypes observed in the leaves as white, green and variegated, that is patches of white and green on same leaf. The progeny derived from the seeds of the white and green plant gave rise to white and green respectively, irrespective of the kind of pollens used from 3 phenotypes. The variegated plant gave rise to all 3 phenotype branches and leaves irrespective of the phenotype of the pollen plant. The phenotype of the seed primarily depends on the phenotype of the plant that produces the egg, and the genes responsible for the phenotypes are present in the chloroplast. The chloroplasts are transferred from the mother to offspring through cytoplasm of the female gamete, and male gamete has no role in transmission of chloroplast DNA. In 1908, Carl Correns provided the earliest example of inheritance linked to chloroplast transmission. Correns discovered a variant of the 4 o'clock plant in which some branches had white leaves, some had green, and some had variegated leaves. The completely white leaves and the white areas in variegated leaves lack chlorophyll that otherwise provides green color. Correns was curious about how inheritance of this phenotypic trait occurred. Inheritance in all possible combinations of crosses is strictly determined by the phenotype of the ovule source. For example, if the seeds representing the progeny were derived from ovules on branches with green leaves, all progeny plants bore only green leaves regardless of the phenotype of the source of pollen. Correns concluded that inheritance was transmitted through the cytoplasm of the maternal parent because the pollen, which contributes little or no cytoplasm to the zygote, had no apparent influence on the progeny phenotypes. Since leaf coloration is a function of the chloroplast, genetic information either contained in that organelle or somehow present in the cytoplasm and influencing the chloroplast must be responsible for the inheritance pattern. It now seems certain that a genetic defect that eliminates the green chlorophyll in the white patches on leaves is a mutation in the DNA housed in the chloroplast. Correns reported that flowers on green branches produced only green offsprings, regardless of the genotype and phenotype of pollen parent, and likewise flowers from the white or pale branches produced only white or pale seedlings regardless of genotype and phenotype of pollen parent. The plants developing from the white or pale seedlings die because they lack chlorophyll and cannot carry on photosynthesis. Correns further reported that flowers from the variegated branches yielded mixed progeny of green, white, and variegated plants in widely varying ratios. The irregularity of transmission from variegated branches could be understood by considering cytoplasmic genes, that is, the plasma genes of plastids. A study of the egg during oogenesis in Mirabilis reveals that the ooplasm contains plastids like cytoplasm of other plant cells. If the egg cell is derived from green plant tissues, its ooplasm will contain colored plastids. If derived from white plant tissues, its ooplasm will contain white plastids. If derived from variegated tissues, its cytoplasm may contain colored plastids only, white plastids only, or a mixture of colored and white plastids. A study of the pollenogenesis, however, reveals that pollen contains very little cytoplasm, which in most cases is devoid of plastids. Without the plastids, the pollen cannot affect this aspect of the offspring's phenotype. It is thus clear that variegation is determined by agencies transmitted through the female and that it is not influenced by the type of pollen used. Now, at last, let us discuss the chloroplast variegation in Chlamydomonas. The unicellular green alga Chlamydomonas reinhardti has provided an excellent system for the investigation of plastid inheritance. This haploid eukaryotic organism has a single large chloroplast containing about 75 copies of a circular double-stranded DNA molecule. Matings that re-establish diploidy are immediately followed by meiosis, and the various stages of the life cycle are easily studied in culture in the laboratory. The first known cytoplasmic mutation, streptomycin resistance in Chlamydomonas, was reported in 1954 by Ruth Sager. Although Chlamydomonas' two mating types, that is, MT positive and MT negative, appear to make equal cytoplasmic contributions to the zygote, Sager determined that the streptomycin resistant phenotype is transmitted only through the MT positive parent. Reciprocal crosses between sensitive and resistant strains yield different results depending on the genotype of the MT positive parent, which is expressed in all offspring. One half of the offsprings are empty positive and one half of them are empty negative, indicating that mating type is controlled by a nuclear gene that segregates in a Mendelian fashion. Since Sager's discovery, a number of other Chlamydomonas mutations that show a similar uniparental inheritance pattern have been discovered. Following fertilization, which involves the fusion of two cells of opposite mating type, the single chloroplasts of the two mating types fuse. After the resulting zygote has undergone meiosis and haploid cells are produced, it is apparent that the genetic information in the chloroplast of progeny cells is derived only from the empty positive parent. The genetic information originally present within the empty negative chloroplast has degenerated. The inheritance of phenotypes influenced by mitochondria is also uniparental in Chlamydomonas. Chlamydomonas reinhardti is a unicellular flagellate that exists as two mating types, termed plus, that is empty positive, and minus, that is empty negative. Each Chlamydomonas reinhardti cell has a single large chloroplast that contains 80 copies of a circular 200 kilobyte genome. The chloroplast DNA is organized into 10 discrete nucleoprotein bodies in the stroma termed nucleoids and is replicated continuously during the mitotic cell cycle. In nutrient-replete conditions, vegetative cells divide mitotically, but when starved of nitrogen, they differentiate into mating-competent gametes. When gametes of opposite mating types are mixed, they fuse rapidly to form diploid zygotes and almost immediately begin a differentiation program that leads to major structural and cytological changes. These changes include deposition of a thick wall, retraction of flagella, nuclear fusion, and fusion of the chloroplasts contributed by the two parents. A key event with respect to chloroplast inheritance is the degradation of empty negative derived chloroplast DNA within the first few hours of zygote formation prior to chloroplast fusion. After a maturation period in the dark which lasts for approximately 5 days and reintroduction to light and nutrients, zygotes undergo meiosis and germinate to produce 4 haploid progeny that can re-enter the vegetative growth cycle. That is all about today's lecture. Hope you have understood it very well. Thank you very much and have a good day.